So I'm going to talk to you today about Bachelor Sperm whales in the Bahamas. The results of what I'm presenting are from a multi-year study. It involved a lot of other people, and we used many, many methods, which I'll go through. And it also included 5 other species, but I'm just going to talk about sperm whales today. Okay, so sperm whales are the iconic whale that most people think of, made famous by Moby Dick. And the way that we identify individuals here and everywhere is from the nicks in the trailing edge of the fluke, which is their tail. They present this when they go on a deep dive looking for food. In the Bahamas, we have in our catalog 247 known individual whales. So those whales are resident here. They're here all year. And typically, if we go out looking for whales, we know where to find them. We will find animals that we have in our catalog. We've also had resights of individuals over 2 decades, so we know the animals quite well. The way that sperm whales structure themselves, similar to killer whales, the mother and her offspring, particularly her female offspring, will remain together forever. So these are called matrilines, and multiple matrilines may associate and form what's known as units. Everyone in those units speaks the same dialect. So I study particularly whale sounds, and sperm whales have really interesting vocalizations. Units may also temporarily associate with other units only if that other unit also speaks the same dialect. And units are stable across decades. The males leave the matrilines when they're subadults because as male sperm whales grow, they become about 1.5 times the size of a female. So they're massively sexually dimorphic, and basically they need a lot of food. And males end up spending the majority of their lives pretty solitary in the polar regions where there's more food, and then they return to tropical latitudes temporarily to mate. I just want to briefly touch on ambergris because it's such a hot topic in the Bahamas where we like to make a quick buck. And I just want everyone to know how rare it is, because we get on average 2 products a month being sent to us, or people arriving at our center hoping that they've found ambergris, and in 25 years it's never been ambergris. So sperm whales eat squid, and squid have a hard beak in them that's similar to the beak of a parrot. And typically what they'll do is they'll vomit up the beaks after they've digested the squid, but sometimes the beak will pass through the digestion system of the whale, and the whale builds up a product around the beaks to protect itself, and over several years, the product builds up and builds up until the whale defecates that, and that is ambergris. So only about 1 in 100 sperm whales ever produces ambergris, and usually only once in their life. So although it's a very valuable product used in the French perfume industry, and it has been found on the beaches of the Bahamas, I wish you luck. So the question that some of the results of our study has helped us to answer is, what are the males doing between leaving their mothers and arriving as adults in the polar regions? It's very unknown anywhere in the world, this period of their life. We know that males leave their mother anywhere between the age of 5 and 15, and then they remain in similar environments, so similar latitudes, until they reach puberty, which is about 19. It's an age of accelerated growth for them, a really important time, and at that time they really start spending more of their life in the polar regions. So this is where we are, and the cluster of, so all the yellow dots are sightings of sperm whales. The cluster in tongue of the ocean next to Andros Island are because we do a lot of work at the Navy base there. So there's a US Navy base called the Atlantic Undersea Test and Evaluation Center. They have 93 hydrophones on the floor of the ocean that are spaced 4 kilometers apart and provide them this excellent laboratory to do a lot of tests with sound, basically, so detecting submarines, torpedoes, etc. And we work there because we are looking at the effects of the sounds on the animals. So that's why there's a cluster there. There's a cluster off southwest Abaco Island because that's where we're based. But typically, they're throughout the canyon. So I discussed that we have multiple methods. So this is how we typically work in an inflatable boat. We take, obviously, photo ID. And for this project, we were using satellite telemetry tags, small limpet tags that stay on the whale for about a month. Ironically, they're a lot smaller than the manatee tags. And we take biopsy samples for genetic and chemical marker analysis. And we also take recordings of the animals. So on big ships, we use a big hydrophone cable that tows a long way behind the ship engine to avoid the noise. And on small boats, we use a handheld hydrophone. For the purpose of this study, you just never know if it's done it. We were using the recordings not to look at the dialects that I mentioned earlier, but just to look at the size of the animals. So sperm whale heads are allometrically related to the total length of the animal, which we know from whaling data. And the way that we figure out the length of the head is because when a sperm whale clicks, which is the sound they make to find food, and also patterns of clicks are their communicative sound as well. The click goes out the front of the sperm whale head, but some of the noise actually travels backward through the spermaceti organ, which fills their head and where they got their name, and echoes off the back of the skull and then comes out the head slightly after the initial click was made. It's an echo. In the bottom picture here is a waveform, and the two patterns of lines, the first one is the click and the second one is the echo, and they're called pulses. So we know the time between the pulses, and that time is 2 times the length of the head, going at the speed of sound in a spermaceti organ, which is very similar to the speed of sound in water. And that's how we can then get the size of the animal. And this has been proven many times over the years as a successful method. So this table shows us the sizes in order. The males are in lighter blue, and on the right, the F and M are when we also had genetics that confirmed the sex of the animal. So as well as knowing ages, we also know from the literature that animals, we know they leave by the time they're 15 years old, and by 15 they're typically 11 meters in length. So here we have a male that's 9 meters. It's still in its maternal unit. That makes sense with the literature. We also have a male that was measured across 1 year and grew almost a meter, and it's at the size of 12 meters, which is about the size that they should have left the maternal unit, and also in that accelerated growth period. And a meter is quite a large growth period. We can also see that the largest animals here are males. It's very difficult when you're in the field to know, one, how big the animal is, especially from a small boat, and also there's this overlap between the size of females and subadult males. So unless you're presented with something obvious, it's very difficult to figure out. Okay, so from the genetic analysis, we found that there was significant differentiation, both in mitochondrial DNA and nuclear markers, between the animals we were finding in Northwest Providence Channel and the animals on the Ortet Range. And when we looked at the photo ID in accordance with this, we found that most of the animals in the tongue of the ocean seemed to be males. When we looked at our tag analysis, we could also see that the males here in blue, the females are in red, and the unknowns are in green. The animals we tagged on the Ortet Range in tongue of the ocean were also mainly males, and they had this much larger ranging pattern in and out of tongue of the ocean than the females, who display this really high site fidelity between Grand Bahama and Southwest Abaco. We did some association analysis. This is it's a typical dendrogram, and what it does is using photo ID, it groups animals together who have been associated over time, and these are called units. So in the Bahamas, we have 7 units, and the mean index within units of association was 0.2 and 0 between, and there's a genetic basis for being in a unit because the animals within units were significantly more related, which fits in with the hypothesis of matrilineal groups. None of the animals in the units were found in tongue of the ocean. These were all the matrilineal units that were found in Northwest Providence Channel. And when we looked at the stable isotope results, we could see that the males, so typically those animals in tongue of the ocean, had much more variation in either their environment or their prey, or potentially both, than the females. So then the most interesting result came, which was that the males that we were finding in bachelor groups in tongue of the ocean were more related to each other than to the females in the matrilineal groups, suggesting they may be immigrants. So we had always thought when we saw these males in tongue of the ocean, they have a different behavior, and initially we thought it was because of the Navy. But they were different to the females in Northwest Providence Channel who, really weren't skittish around the boat at all. But we thought these males were in the process of moving away from their matrilines, but that they had come from Bahamian matrilines. And what we've learned is they haven't. They've come from somewhere else. But really interestingly, as they've come together, they're related to each other. So this has never been known before. We don't know where they've come from, but we're going to work with some colleagues, particularly in Dominica. And some people have already talked about birds not having boundaries, but the same obviously goes for whales. Unfortunately, the area that the animals have chosen to stay in for a year or two on this journey is in tongue of the ocean, which the females basically never go to, presumably because of the insonification of that water. So we tagged 19 sperm whales in tongue of the ocean, but only one animal was tagged and present during a multi-ship exercise that uses a lot of sonar. This chart on the left is temperature, so at the top is much hotter, so surface, and at the bottom is much cooler, representing depth, and on the x-axis is proportion of time. So the animal that was exposed is in red. It spent about half of the time compared to the non-exposed whales at the surface, and about 3 times the length of time at depth. When we modeled the speed of the animal at the time of the exercise, you can see that it went down the left-hand side of tongue of the ocean, it foraged in the cul-de-sac, and then when the exercise began, it sped up going as fast, the fastest time it swam during the time it had its tag on, up as far to the east of tongue of the ocean as it could, and straight out of that body of water. So in conclusion, we have some bachelor male groups in the Bahamas. They appear to be immigrants from elsewhere, roaming together, but they may be repeatedly disturbed at Alltech during a really important growth period in their lives. Thanks.